The cells in the nervous system. Neurons vary in size and shape. They differ in length, the size of their axons, the size of their dendrites. But all neurons have three main features: the cell body, or soma, which contains the nucleus, the cytoplasm, the organelles, neurofilaments, and chromatophilic substance, used to be called Nissl body. Those neurofilaments and chromatophilic substance are specific to neurons. Dendrites are receptive surfaces, and many neurons have many dendrites on them. Axons transmit impulses and release neurotransmitters to an effector organ, and neurons generally only have one axon, although that one axon can have branches that would make collaterals. The cell body contains the neurofilaments and the chromatophilic substance. Here, there are dendrites attached to it. The dendrites include the region called the axon hillock, which leads to the axon, and the axon can have branches, which are called collaterals. They have a terminal, and on the end of that terminal is a synaptic knob, which makes up part of the synaptic cleft, which we talked about last week, and we talked about muscles. In the periphery, those neurons are also insulated. Many of them are insulated by myelin, which is provided by a myelin sheath, which is produced by something called Schwann cells. Those Schwann cells are found only in the periphery. They're PNS neuroglia, encase the axons in a sheath. They wrap tightly around the axon in layers that are composed of myelin, which is a lipoprotein mixture, lipids and proteins together. And that coating is called the myelin sheath. The spaces between the Schwann cells are known as the nodes of Ranvier, and they allow for, they are not myelinated in those points, and that's important when we talk about saltatory conduction in myelinated axons. They're not all myelinated. The myelinated ones have a series of Schwann cells lined up with gaps between them, producing myelin and wrapping around that neuron, whereas the unmyelinated axons, while they have Schwann cells with them, those Schwann cells are not producing myelin. That's important because of a disease called multiple sclerosis, and in multiple sclerosis we get destruction of the myelin sheath in the central nervous system. In the central nervous system, it's not produced by Schwann cells, it's produced by a different cell. That myelin is attacked by your own antibodies. There are scars or scleroses that are left behind, and that's why it's called multiple sclerosis, because there are many of these. And when you have them, those will stop neurons from conducting impulses. Without impulses from neurons, muscles stop contracting and atrophy. Many other things can occur, including fatigue, mood problems, blurred vision, weak, numb limbs. Treatments involve drugs that suppress the immune activity. Neurons themselves can be classified by their structure. We have multipolar neurons, which include almost 99% of neurons. They have many processes, most of which are dendrites in a single axon. Most of the neurons within the central nervous system are multipolar neurons. We also have bipolar neurons, which have two processes, one axon, one dendrite. And those are important in some of our special senses, like our eyes, our ears, and our nose. And then there are unipolar neurons, which have only one process. Their cell bodies are in structures called ganglia. They have a connection, axon-like connection, and a dendrite-like connection. And they are important for sensory information. We can also classify neurons by function. So sensory neurons are afferent neurons carrying impulses toward the central nervous system. Most of those are unipolar, a few are bipolar. Then we have neurons that connect things within the central nervous system. They are also called association neurons or link neurons. They are multipolar, and they are located within the central nervous system. And then we have motor neurons, which are multipolar, efferent, carrying impulses away from the central nervous system. carrying impulses to the effector organs, skeletal muscles, or glands. Neuroglia. The general function of neuroglia is to provide structural support for neurons. In the embryo, neuroglia helps guide neurons into position and may stimulate their specialization. They produce growth factors to nourish the neurons and remove excess ions and neurotransmitters, and they help in the formation of synapses or connections between neurons. In the central nervous system, we have astrocytes, which connect neurons to blood vessels, exchanging nutrients and growth factors. They are also what forms scar tissue when there is injury in the central nervous system. They help to metabolize certain substances, and they regulate ion concentrations, such as potassium ion concentration. They are a big part of the blood-brain barrier. Oligodendrocytes are going to myelinate central nervous system axons. And unlike Schwann cells, many Schwann cells cover a single neuron, one oligodendrocyte can cover several different neurons. Microglia are phagocytic cells similar to macrophages, but they also provide structural support. And in the central nervous system, we have epidermal cells, which line the central canal of the spinal cord and the ventricles of the brain and cover our choroid plexuses. And they are very important to help regulate the composition of cerebral spinal fluid. They are cuboidal or columnar cells, and they are ciliated to help move the central cerebral spinal fluid around. In the peripheral nervous system, we have two neuroglia cells, Schwann cells, which produce the myelin sheaths in peripheral axons. That is going to speed up nerve transmission by allowing for saltatory conduction, which we'll talk about later. And satellite cells, which are clusters of neuron cell bodies or ganglia clusters of cells that support those neuron cell bodies or ganglia. Mature neurons do not divide. They are amitotic. If the cell body itself is injured, a neuron usually dies. In the peripheral nervous system, if just the axon is injured, it can regenerate itself on occasion. The axons is separated from the cell body, its myelin sheath degenerates, then Schwann cells and neurolemma remain, and those Schwann cells provide a guiding sheath for the growing axon. If the growing axon establishes the same former connections, the functions will return. If it does not, the function may be lost, and it takes a great deal of time for those connections to be remade. Within the central nervous system, the axons do not have neurolemma to act as a guiding sheath. Oligodendrocytes don't proliferate after the injury, and we have very poor evidence that we get any regeneration at all within the central nervous system. Okay, I'm going to stop there.